So before we start, thank you for joining the first edition of the 1 Hour With. And this is the very first edition. Some rules before we start. So you can ask questions. You have a module at the bottom of the screen. If you want to ask question, just type your question. I'm going to take them at the end after the interview. And we try, to answer all the questions. We're going to have a recording for the session, and most probably we're going to put that, as a podcast, so you'll be able, to go back to it, and it's going to be available on our website. So it's time. Welcome, everybody. Good afternoon, good evening, good morning. For most of you, I saw that we have friends from Canada as well. So this is the very first edition of the 1 Hour With. Welcome to Pierre Rocheteau, he's the CEO from Olgram. As you know, seaweed is a very interesting topic, not only for the environment, the nutrition, the cosmetic, but also for the pharma industry. And Pierre is here today on the hot seat to try to explain what Olgram is aiming to. And he will share his vision, his ambition, and some interesting directions where the seaweed could be used in the next coming years. So Pierre, as an introduction, I'm going to give you the floor. You can share and start sharing your presentation. We're going to have 10 minutes of the global presentation. Then after, we're going to jump into the discussion and the interview. Thank you and welcome, Pierre.
All right, so I'm going to share my screen. Here it is.
Perfect.
So, perfect. All right. So hello everyone. Thanks for joining this talk. Before I start, I would like to thank Pierre and the entire BioMarine team for giving me the opportunity to present Olgram. It's a great honor to be the first one. Thanks a lot for this. So I'm gonna introduce you to Olgram. It's a very early stage biotech company. We created the company last year. So I'm going to introduce you the company and our vision. So in four bullet points, what is Olgram? Olgram is a preclinical stage biotech company with unique first-in-class molecules. We have a diversified portfolio of innovative compounds, both in immunostimulation and infectious diseases. We have two different programs. The first program is a new molecule found in algae able to boost the immune system of immunodepressed patients. And I will, of course, come back into this program, which is of main interest for this audience. But bear in mind that we have another program, the program number 2, where we have found in the sea a molecule able to kill persistent bacteria, which are responsible of chronic infectious diseases. And we are the only one in Europe to have this kind of molecule. But this one I'm not going to talk about. We can discuss later if you want, which will be my pleasure. So as I told you, we have two programs. They are at different stages. One is at the lead optimization. The other one is just starting the lead optimization. And as I told you, I'm only going to describe the second program, which is new molecule found in algae to fight against immunodepression. So before I jump into the project, I would like to introduce you the founders. One of the founders is Hervé Balusson, the chairman and founder of the Olmix Group, who is a group working on transforming algae into many different products in plant care, human care, and animal care. Marie Van Jans, she is the chairwoman of Olgram. She is also the founder and the strategy director of OSE Immunotherapeutic, a publicly traded company on immunology. And she has already created and sold many biotech companies. Myself, so I am Pierre Rocheteau. I'm the CEO of Olgram. I have a PhD from the Pasteur Institute on infectious diseases and stem cells. There is a link, believe me. And I also have an MBA from ESCP Europe, and I was in Mr. Balusson's human care department before. Professor Karima Zenoun is the head of anesthesiology of the Nantes University, and he already conducted 27 clinical trials on the subject that we are interested in. And the Dr. Pinival Collen, she's the scientific director of the Olmix Group, and she knows a lot about algae and the algae themselves, but also the industrial process to extract molecule at the industrial level. So As most of you have probably already known, because you all are in the algae field, we are talking about marine sulfated polysaccharide that are known in algae to have a broad pharmacological effects. They have anti-inflammatory, anti-angiogenic, bacterial, coagulant, diabetic, antiviral, gastroprotective, and immunomodulatory effect. There are a lot of papers describing this, so this is not new. And we know that there are many MSPs in algae, many different MSPs with different properties. Well, we have found and selected one with original and unique biological properties. And this is what I'm going to show you. So that's how the story started. It started in the Olmix group and Amadeus company, where they have shown that a marine sulfated polysaccharide, an MSP extracted from ulvan, an algae, had very interesting effect on chicken and piglets. Here you have the results for the chicken. You can see the more you give the MSPs, the algae, the more the immune system of the animals are active. And this was a very, very interesting property because it allowed the reduction of use of antibiotics, but I will come back into that probably later. So from this observation, what about the use of those MSPs in the context of immune depressed patient, as we have a molecule that is able to activate the immune system? So we decided to focus on the immunodepression after traumatic brain injuries. What's going on after a traumatic brain injury? You have your brain here, and if in case of a trauma, for example, after a car crash or after a surgery, the brain will send, because of inflammatory mediators, will send many signals, and these signals in the end will tell the rest of the body to decrease the level of activation of the immune system. And this is translated by a decrease of IL-12. I'm not going to enter into the details, but this is our readout. I will come back into that later. Another way to see it is this way. You have a trauma. You have, of course, an activation of the immune system because of the trauma. And after this activation of the immune system, in all the patients, you have a decrease, a collapse of this immune system. And most of the patients, 99% actually, do survive because the immune system can go back to normal. But in 11% of the patients, you can see that the immune system stays low. And a low immune system at the hospital is really bad thing because of nosocomial infections, viral reactivations, and ultimately, a lot of patients die from it. So what we are talking about trauma, immunodepression post-trauma. We have 5 million patients with traumatic brain injury in the US and Europe. 15% of the patients end up in ICUs, in intensive care units, and 41% of those patients do get pneumonia. And there is a need for an immunostimulatory molecule to boost the immune system in immunodepressed patients. It will be a first-in-class molecule. There are no molecule able to boost the immune system in this condition. So that's what we want to do, and that's what I'm going to show you as preclinical data. So a little bit of science. I have 3 slides of science. Stay with me. First of all, I would like to tell you, MSPs are not an anti-infectious. You can add MSP in vitro or even in vivo. You will not destroy the bacteria. It's not an anti-infective. However, and I'm going to spend a little bit of time on this panel because it's very important. There you have the immune system of naive, normal, classical animals. When you inject a bacteria in the lung of an animal, you have an activation of the immune system, of course. But when an animal is traumatized in the brain and you inject a bacteria, you can see that the immune system does not activate, as I showed you before. And when you inject the bacteria with the animal traumatized, but with MSP, you can see that the immune system can be again activated. So the MSP are able to reactivate the immune system. This is really a key result. It's really our key result. So very quickly, another experiment, another readout that we were not expecting at all is the fact that there are no dissemination of bacteria when we inject the MSP. We can discuss about this later, but when you inject bacteria in the lung, the bacteria stays in the lung for at least 3 days when you inject MSPs, whereas when there are no MSPs, bacteria go all over the body. And what's also very interesting is that in naive animals, MSP do not trigger immunological reaction. So we have a mechanism of action. I'm not going to enter into the details. We can discuss science later if you want, with pleasure. But we have a molecule that boosts the immune system, only when there is an immunodepression, and that avoids the dissemination of bacteria. And it's those 3 properties that we are now exploiting. So this also is very open for discussion, but probably you know this. We have other results that are very, very interesting. For example, the MSP, so it's not exactly the same MSP, it's another fraction, but still it's an MSP. We have shown in our hands that those MSP were able to act as an antidepressant. We will come back to that later in the discussion, but we clearly have a molecule that has an antidepressive activity. And these are not our results, it's taken from the literature, but you have probably seen that already. Those MSP have a very interesting effects on the COVID, on the SARS-CoV-2 in vitro. It has been shown also in vivo, and we have results showing that it's active on other strains of viruses. So what is our ambition? What do we want to do? So Olgram, we want to culture, extract, transform at the pharma grade the MSPs. We want to characterize, generate as many fractions as possible with different biological activities. So we have our MSP, we'll have many fractions of MSPs with different biological activities. And internally, we'll go through the clinical studies in order to do the drug development for the immune system in the context of traumatic brain injury. But we would like to have as many partnerships as possible in order to develop the other clinical applications, as we will have the MSP at the GMP pharmaceutical grade level. But we also have other opportunities in non-clinical applications, such as nutraceutical, but that you also know better than me. So this is my last slide. This is a report taken from the Boston Consulting Group, published in 2020. There are many challenges in the pharmaceutical industry, and I think I want to show you why Looking in the Sea will be creating a winning biopharma strategy in the 2020s. So first of all, we must master the new logic of competition. This is something the big pharmaceutical company do very well, but biopharma is grounded in innovation, and we need really to leverage new and old data sources. We have to be state of the art to, for example, modify genetically algae using CRISPR-Cas9 of this kind of technology. But we also must master perfectly the old techniques of culture. We must master perfectly the old techniques of biochemistry to extract molecules, and so on and so on. And we really need to focus, not give up on the old way of getting data, but having both the new ways and the old ways. Designing the company of the future, it requires a cross-sector collaboration. You probably know that from culture of algae to making an intravenous molecule to boost the immune system, it requires a lot of different skills. And we need, as a result, to have these different within the company, within Olgram. And this leads us to the next point, which is to embrace the need for diversity. We need to apply the science of organizational change. We can come back to that later, but the world is changing very fast, especially this last year, which has been very special, and we need to embrace this. And in the end, optimize for both social and business value. Why? Because the patient at the hospital doesn't care that the molecule is from algae and the molecule is natural. He's dying, so he wants something that works. However, the general people, they do really care that we start to do things in a sustainable way. So this is something we should start to take into consideration. It's a very, very strong wave that is coming. So this is why I was proposing a kind of purple biotechnology concept, because you have the red biotechnology, pure players in the health and the clinics biotechnology. You have the C, the deep tech, the blue biotechnologies. You know that. What is the blue biotechnologies? We have to merge both. We have to be in the purple biotechnology. We need really to merge those health and blue biotech sectors. And if I believe what says the Boston Consulting Group report, by doing this, we could be creating the winning biopharma of 2020s. And this is what Olgram will do and is about.
Thank you very much, Pierre, for this quick introduction. Many questions, I guess, and I'm going to check online. Pretty sure that some of the participants will have to dig into more details. But the purpose today is really to understand what is behind the science, what will be the delivery, from the company. And before we get to that point, let me rewind the story. I'd like you to explain in a few words what is the idea nested and the history and the relationship with the Olmix group to get into the context.
Yes, of course. So I have a brief slide about our relationship with the group. Here it is. So a few words about the group to start with, to understand what are our relationship with the groups. This slide is about the Olmix group and also the company Amadate of Mr. Hervé Balusson. So the group is cultivating and harnessing algae, and they are transforming algae in different sectors. In the plant care, for example, they are making fertilizers and pesticides that are natural. In the animal care, they are making supplement, dietary supplement to boost the immune system of the animals, and they also had a human care department. And the project started this way. They realized by doing a lot of experiments with INRA and other universities that when you give an extract of algae to the animals, there is a boost of the immune system. And as a result, with this boost of immune system, you can avoid the use of antibiotics. Look at that. Those results are always amazing. You have a 92% decrease of use of antibiotics when you put algae in the food of chicken in that case. And I have shown you those data very quickly before with the boost of immunostimulation. This is amazing. And that's what the Olmix group is doing. They are making food, healthy food without antibiotics. They help doing this. And Mr. Balusson thought, well, okay, if it works on animals, is there a way for this molecule to be active or to be used at the pharmaceutical level for human beings that have problems of immunodepression? And the group did a lot of exploratory experiments with INSERM, with the University of Nantes, with other CROs on very many different clinical applications. And in the end, we decided that the most interesting one was the experiment discovered within the University of Nantes, the Hospital of Nantes. And at one point, we already had this discussion a lot of time. At one point, immunodepression, you have a lot of cases where you have an immunodepression. It can be environmental, it can be age, it can be surgery and trauma. I discussed about that. It can be malnutrition, HIV, genetic, metabolic, or immunosuppressive drug, for example, for chronic inflammatory disease or cancer even in chemotherapy. So we had to choose which was the best applications, and we have data on many of those aspects. And the most interesting one, both at the market, both at the medical need, and both in terms of result that we already had in-house, was on the trauma. Because trauma, there is absolutely nothing, and there is a huge need of boosting the immune system. That's how the project originated.
Pierre, another side question, and I'm going to start to answer some of the questions that are coming, live. Just to come back to the context, Olmix started with ulva. We're talking about ulva and green seaweed, correct? But when you mention MSP, marine sulfated polysaccharide, it could also be found in different species. So is it a broad scope that you envisage in the future, or is it, very focused on ulva and, building up on this unique property that the ulva has?
Yes, we will build up on this property that ulva has. We have done a lot of experiments in the past. First of all, to select the good seaweed, the good algae, and we did a lot of experiments in different kind of algae. We also, within ulva, tested different fraction, because MSP, I told you, you have many MSPs with different sizes, different configuration, different level of sulfonation, all those things were tested. And that's why it took us so much time, because this is an idea that was nested in 2016, but we needed really to test a lot. And in the end, we came up with this property of MSP, a specific MSP with a specific size, with specific level of sulfonation in ulva, that has the most interesting property. And now, as you have seen, we really want to focus into that. We stay open with other clinical applications because we have something that boosts the immune system. However, the nature of the molecule, maybe we will modify it via lead optimization, but it's going to be a chemical thing. It's not going to be the species of the algae that will change.
We all understand, the interest and the trendy aspect of this seaweed, because everybody wants to get into seaweed to make cosmetic, advanced product, biomaterial. But I'd like now to understand better why you decided to choose this very challenging niche, because when you shoot for the star, and pharma is, you know, almost the moon or the sun, it takes a long time to develop. So can you, in this context of the pandemic, maybe comment a little bit more on the program that you have, and why you choose, you know, the brain injury instead of IBD, which is also very interesting, anxiety, or, you know, tell us more about, you know, the different lines that you're developing, and elaborate maybe a little bit more on the real potential of this extract for this type of new drug candidates.
Yes, yeah, with pleasure. Well, keep in mind that historically, the very first experiments were done in the Olmix group. And the Olmix group is treating plants, is treating animals. They are doing cosmetics and things like that for human well-being. And to close the loop, health was the last things we wanted to tackle. So this is how we started to be interested in human health. This is just a quick historical point of view. You told me regarding COVID and regarding the situation. Well, there is a huge situation right now that we need to tackle now. It's this one. Probably many people know this. I'm sorry, we are in the middle of a pandemic. I don't like to have this kind of talk, but I have to. By 2050, we will have 10 million deaths due to antibiotic resistance. The Olmix group is fighting against this. But just to make it clear, 10 million death means 20% more death than cancer, and cancer in general. I mean, all cancer. So it's a huge number of deaths. Why? Because today antibiotics starts to not work anymore. This is a huge problem. So we have solution in our hands. We have a molecule that can be used at the hospital to boost the immune system that would allow us to avoid the use of antibiotics at the hospitals. Because when you discuss with doctors in the ICUs, they are not using automatically antibiotics anymore when the patient go in the ICU. They used to do that a lot in the US, a little bit in Europe, and they have stopped everywhere. You cannot use antibiotics when a patient has a trauma brain injury, because if you wait a year, you will have huge level of antibiotic resistance in the entire service. So they have totally stopped to use the antibiotics. So they desperately need a molecule that would avoid the use of antibiotics. And in the context of COVID, well, we thought this problem will arise in 2050, but because of COVID, it will arise much earlier. People have been using a lot of antibiotics to treat a virus, which doesn't work at all. And people have been using much more antibiotics than they should have. And we know, we don't have a figure yet because we are still in the middle of the pandemic, but we know that this problem will arise much sooner. Maybe not stronger, but sooner for sure. And we are in 2021. It takes 8 to 10 years to develop a medicine. And we are not going to go from 700,000 to 10 million in 1 year. It's going to be gradual. So we will probably have between 5 to 6 million deaths by 2030, 35. So it takes 8 years to develop a drug. We need to do it. Yeah, I know. Sorry, I don't like to have this kind of talk, but it's a reality. So we absolutely need to find new ways to do that, and we wanted to have an impact and decided to tackle the pharma industry.
That's very important. I'm a bit scared, 6 to 10 million. So it's yeah. I hope in the context of, this pandemic will help us to improve, this new drug delivery and these new candidates. And just coming back, to this fast track, I would say, in discovering vaccines and new candidates, is it important, to consider synthetic biology or CRISPR as a key vector for improving, the performance of the drugs or not?
Yes, this is something we are considering. Now that we have the characterized the MSP, that we know which molecule is very active, we are wondering if there is not a way to modify the algae with CRISPR in order to boost the production of this specific MSP. This is something that is ongoing. it's really exploratory. So it's not in the main pipeline of our development. But at one point, if we have to switch from one algae to a GMO algae, that's not going to be much of the change at the step where we are standing now, because later it's going to be a big deal, of course. So the project is still ongoing. Now we are really focusing on two separate aspects of the project that we will merge at one point. The first one is, well, we have a molecule. We have to do everything that needs to be done at the pharma level, understanding the pharmacodynamics, pharmacokinetics, understanding the absorption, distribution, metabolism, excretion, toxicology. All those aspects are ongoing. And of course, how to provide the molecule. Right now, we have an algae. We know how to culture. We know how to extract. We are adapting the protocol to be at the GMP pharma level. But we also have a project ongoing where we would like to modify the algae with CRISPR-Cas9 and have a GMO to overproduce this specific MSP. That will be a very, very good thing to boost the yields and to have a much cheaper way of obtaining the algae.
Pierre, I'm going to start, to take some questions because everybody,
please.
has, lots of questions, and I think it will be relevant also to have them to better understand. Coming back maybe to the raw ingredient, we have a few questions that are raising about, the type of quality you need, because most of the people, they know very well, seaweed production. Everybody wants to get into this, but when you want to reach, the pharma grade, would you say that it's much better to have an RAS system, which is very content, very efficient in terms of quality, or can you also use natural organic seaweed that grow in the sea? So
there is no way we can use natural algae in the sea. It's not possible, because at one point you need a genetically pure strain of algae. So you cannot take the algae in the sea. And our MSPs, what's very important in the pharma industry is that we always get the very, very same product. This is very important. When you take your medicine, aspirin or something else, and it's written 1 milligram per whatever, it's always the same drug. You take it in 2016, in 2020, whatever. It's strictly always the same thing. And taking algae from the sea will be very, very difficult to reach a product that is at the nanogram level, the very, very same. So we have two different kind of culture, and it really depends on the country you are considering. The regulatory agencies like European Medicine Agency and FDA are okay with cultivating algae in outdoors, outdoors, but inland. However, they need to have the GMP level pharmaceutical grade, and they need at least 3 steps between the thing you will inject and your raw product at the GMP level. And this is something we will rely on external partnerships to begin with. We will extract our algae, we will extract the powder, the MSP. We will get the MSP as pure as possible, but at one point we will need to give this powder to a GMP grade CRO so that they can make something injectable and reproducible to the patient. Some FDA, we are still, the discussion is still ongoing, and FDA is also considering, because it's not clear-cut yet, of cultivating algae in aquariums.
Is it an FDA or a GRAS that you're looking for? Because for this type of product, you're shooting for an FDA approval.
FDA approval. It's going to be a medicine. It's going to be a drug. So yeah, it's FDA approval, and yeah, we need. Yeah, sorry.
It's not single molecule or mixed molecule, but it's purified extract with GMP quality. And do you have different type of molecule that you're using, or is it a single molecule in the end for the candidate?
So actually, up to now we have 3. We have a fraction with 3 molecules. We know them. They are perfectly characterized, and we will always need to have the same ratio. This is important. Always the same ratio, always the same level of sulfonation, and so on and so on. And we need readouts. We need HPLC readouts for the people who knows a little bit chemistry. We need HPLC readouts, but also we need biological readouts, which means that every batch we need to set up in vivo and in vitro biological readouts to tell us, yes, okay, the activity on inflammation of this batch is X, the activity of this batch is Y, and so on and so on. So we need to characterize perfectly the extract. And now I told you we have 3 molecules. I'm very confident with the fact that we will go down to 1 molecule. We are almost there. I think by April, we will have 1 molecule. And I will be able to tell you, is it a synergy between the 3 that makes what's interesting, or is it really one of them that is really, really interesting? So this I will be able to tell you by April.
Yeah, we will come back to the different question because we have a long list. Let's talk about, the long journey of a marine biotech company. We know that reaching the phase 2 is a long journey, but phase 3 is almost impossible when you are started. So we have some investors in the conference today. What will be the takeaway message? Because are you a technology platform that will help to design the first candidate and then pass it or sell it, or are you more on the long run, and how you envisage this this future development, because it's quite costly?
Yes, it's very costly. If we talk about costs, we will need around 150 million euros to finish phase 3. And that will be not before 7 years or 8 years, depending on how things goes. So yeah, it's very, very costly, but the reward is huge. I mean, if we reach our goal, we will be a first-in-class molecule where all traumatized patients will use, and the price of this kind of product can be very, very high. We've done the medico-economic study; it can be very high. So it's risky, but very rewarding. So that's my first message. Our goal is to go at least to the end of phase 2. To reach the end of phase 2, we will need around 35 million euros because it's phase 3 that really costs a lot. And at the end of phase 2, we have 2 options: either partnerships with a big pharmaceutical company with exclusivity, or maybe the stock market. And this is in general, if I discuss with investors, this is in general where people start to envisage an exit, a first exit. You can exit at the end of phase 2 because we have a positive result. And if you want numbers in the infectious disease, the probability of success when you have reached a successful phase 2 is around 70%. So it's a high successful rate between 2 and 3. You still have a long way to go, but it's a high rate of success. So we want to go at least at the end of phase 2. That's the minimum we want to do. There's a first exit there, but if we can push it to the end of phase 3, that's where you make the most of the value, of course.
Okay, so you mentioned the 35 million that you're going to raise, to reach, the end of the phase 2. So far, what have you raised, and what is the next milestones that you want to achieve, and when can we look, at the next round?
Yes, so we just closed 2 weeks ago our first seed round. I told you we were created last year, and we closed the seed round this year. We closed 1.5 million euros 2 weeks ago, and with this money we will do 2 things. I told you we have two programs. For the MSP program, we want to go up to what we call the preclinical data. We want to be done with the preclinical data, which means that we will have a name of the molecule. I will continue to call them MSPs, but I will have an exact complete name, chemical name of the molecule. I will know what's their behavior in an animal, which means absorption, distribution, metabolism, excretion. And I will also have the toxicological profile of this molecule. I will have all of this. With this money, I can go up to let's say the start of the absorption, at the end of absorption, distribution, metabolism, excretion. So I will have a molecule. And also with the other program, I will be at the end of preclinical studies. This is what I do with 1.1 million. But if I want to completely finish the preclinical studies for both programs, I need 6 million euros by the end of 2021. And by the end of 2021, if I get these 6 millions, by mid 2022 or end 2022, depending on how it goes, we will be ready to start a clinical trial. So long story short, we have 1.5 million now, and with an extra 6 million, I will go from preclinical biotech company to clinical stage biotech company.
Is there a way in between to get, a clever industry partnership, not with pharma, but with companies that are investing a lot in customer care or patient-derived, product to heal them and to improve, their health. Is there a middle way where, you're going to have a drug candidate that could be exploited over the counter?
Absolutely. So our focus really is going through from proof of concept, and now we have more than a proof of concept. We have interesting preclinical data to the clinics in the traumatic brain injury and immunodepression. That's really our focus. However, we will also take the opportunities of exploiting the other properties of this MSP. I told you we have very interesting antidepressant properties. It's amazing that after 68 days of taking MSP, 91% of depressed people are feeling better compared to only 60% of people who are taking a placebo. It's a very serious clinical trial that has been done, 82 patients, double-blind, randomized, and so on and so on. So we are absolutely certain about our results. And there is a huge potential for clinical nutrition, sorry. For example, if you ask to a psychiatry hospital and tell them, well, we have a drug that will allow your patient to be out of the hospital sooner because it potentializes an immunodepressant, this would be very, very interesting for the hospital. And this is something they're ready to pay a very high amount of money too, because a patient at the hospital costs a lot. So yes, we will grasp other opportunities. And IBS, you mentioned IBS. It's also very interesting. We have those amazing results in it's another MSP.
Yeah, can you rephrase, IBS? Can you explain?
Sorry. Yes.
Some are investors and they are not very familiar with our, language.
Yes, sorry. So yeah, of course, it's the intestinal bowel syndrome. It's the bowel that's not functioning properly. And we have shown that those MSP, another fraction, is very active and very interesting because it increases the permeability and decreases the intestinal stress of animals. And we have those data, we have patents. And of course, if there is a nutraceutical company that is interested in putting this in a pill, in a yogurt, or and so on, why not? I mean, we are very open to co-develop a product or to sub-license something. We are very open to the way this thing can be organized. They can have MSP in their hand, test in their own lab, and see what we've got. But I can tell you, we have been working with different CROs, and in the antidepressant part, they compared our MSP with actual antidepressant, and they see, okay, we have the same result in the rat, and we have those very impressive results in human. And for the bowel, the intestine bowel, well, they have also amazing results in yogurt or as pills in animals where you simulate a bowel syndrome. So yes, we're going to really focus on the trauma and on the immunodepression, but very open to go for partnerships and sub-license on other applications. Non-clinical applications. Just I want to make a point that is also very important, is that we are not talking about the same molecule, because pharma companies, we have one for immunostimulation post-traumatic brain injury. This molecule is dedicated to this application. And we have another molecule for the Bowel syndrome, another molecule for the depression. This is really key because pharmaceutical companies want their own molecule, and this is normal.
Understood. Not to disclose any confidential information, and we're working together on this, but do you have any offtake, so far, or R&D partnership that you have already started with big pharma or nutraceutical or consumer groups?
Yes, we do. We do have three. Three of them are interesting. They kind of started their due diligence, and I have been very even surprised by classical CROs. I have a project. I'm asking a CRO, well, I need your skills to do this, and I will pay for this service, of course. And one of them told me, yeah, okay, maybe you're not going to pay for this service. Let me investigate if we don't want to invest in the company to give you this service. I was very surprised. I was not expecting this. I know that it's something that they do sometimes, but this project and the other project, I'm sorry, I don't have time to talk about both, but both projects are really, really promising, really, really interesting. yeah, we have very positive feedbacks from the preclinical data that we've got.
Excellent. Let's try to looping the loop, and
Yeah.
I'd like to get more on this side where. So, what will be the need in terms of development, in terms of human resources? Are you staffed already, or what is the plan, for the next 5 years using the proceeds of the fund? What will be the type of recruitment that you're working on?
Yeah. So we have great ambitions, and honestly, we need international profiles now. So right now, what we've got within Olgram is, I told you, Marie-Vonne is chairwoman, I'm the CEO. We have a medical director, Alain Châtelain, and Olivier Favrebull, who is a specialist of regulatory and CMC aspects. This is really, really important because every time we do something, we directly check that it's something that is feasible at the pharma industrial level. Otherwise, there's no point in going further. So all those people are discussing together, and we are doing the project all together. We also have a CFO. We have a scientific team. We have 4 people. Maeva is the project manager in charge of the MSPs. We have another project manager in charge of the other anti-infective program, and we have also a chemist because it involves a lot of chemistry, those projects. But what I want to say is that we have just raised the money now, so we are actively recruiting people now. I want an international CSO profile, scientific officer, a chief scientific officer that will be able to conduct and manage both of those programs, and also a business developer, because I mean, we have so much potential applications from the algae to the drug. At every level, we have a potential application, and I would like one, or even later more, but business developer to go for specific partnerships and see how we can develop these things. Can you imagine, along the way from the algae to the medicine, we have a molecule that is very, very potent in blocking UV light. For the moment, we have not done anything with this property because we cannot focus on everything. We are focusing on traumatic brain injury, but it will be very interesting to know if there is no cosmetic company interested in getting this molecule.
Yeah.
So
and we know a few, so that might be interesting. Yeah, absolutely.
Yeah, I can imagine. So yeah, and I have also, maybe it's the moment to tell this. Sorry, there is a lot of text, but an interesting scientific board. So some people are still XXX because I have not gathered for the first time this new scientific board. So I would like to keep the news a little bit secret until the first board so that I can make an announcement. But we have 3 founders, French founders. I told you about them earlier, but we also have someone from GSK, and which also a panelist from Carbix, the Association of the Anti-Infectious Disease, that knows perfectly at the step where we are, what we do, and also a guy from Merck, and someone in Harvard microbiology who is really specifically working on what we are doing now about chronic infectious disease, and they all are in the board, but can't tell you really now. I'll keep this for later.
Excellent. It looks like you've got a plan, and
yes, we do.
In 2 minutes before we jump into the many questions that we have to answer, I would like to understand better who is Pierre Rocheteau and what is behind you, what drives you, and tell us more about your affinity, the blue bioeconomy, and who you are really.
Who am I? That's a tough question. That's a philosophical question. So I'm to start with, I'm a biologist. I have a PhD from the Pasteur Institute, and I always wanted to study biology because I was amazed. But I wanted to understand how life works at the scientific level, not the philosophical one. I wanted to understand science and how things were working. And at one point during my studies, I realized that studying biology or creating medicines was all about hacking biology, hacking life, hijack life. You have a process. Well, how do I hijack this process in order for someone who is sick to become healthy again, or even to augment people, because this is a subject we cannot avoid anymore nowadays. So this was really, really interesting. And what drove me to the biotech sector is that I saw a talk from Bill Gates in 2005. He was saying that biotechnology in 2005 is the informatic of the 70s. It's something that is in the lab, something people do not really understand. You can see with the Moderna BioNTech development, it went super fast and people are afraid. It's normal. I mean, it's something very new. We have no short delay since the first injections and so on and so on. But really, it's the informatic of the 70s. And when I see all the projects in the biotechnology sector that are ongoing now, I mean, we will be able soon to do a lot with biotech, from light. For example, you can make light with biology. You can make ink. You can reprogram a bacteria to make ink, so there's no need to have those big chemical factories anymore. So I mean, the potential is absolutely huge. The potential is absolutely huge. So this is what drove me to the biotech sector. And last, and that's maybe also very important, is that I always wanted to make a big impact on the world. So I don't care to be famous, really. I really don't care at all. This is not in my way of being. But I want to know that people who are sick at the hospital are using my drug and that they are alive thanks to me. This is something that is a huge motivation. And the last link, the link with Blue Bio sector. I'm from New Caledonia. I lived there for 15 years. New Caledonia is a small island close to Australia for the international audience. It's a very small island, French small island. And so I have been living with the sea every day. So I have a deep connection with the sea. And I mean, it's amazing. We know more about the moon than about the sea. That's what we say. So there's a lot to discover in the sea. It's amazing, the potential of this environment.
Very interesting, Pierre, really. And so let's try, to answer the long list of questions we have. Please. So I'm going to go back, to Sam. Sam is based in Scotland. He's a young entrepreneur developing this amazing concept of recycled aquaculture system. And his question is the extraction method with Olgram, or would you be able to purchase the extract from a supplier?
Yeah, that's a very good question. We would like to master as much as possible from the algae to the drug. At one point, we know that we will need to go through a partner because the GMP level will require an extra 300 million euro investment, which is not feasible at this point. But we want to go from the algae to the MSP pure powder, and this powder will be given to a partner that will transform it into a drug. This is what we want to do. So we will do it in-house. Not sure we will purchase it. In the pharma industry, they really need to have regular, everyday, perfectly standardized processes. And I think it's best to have something in-house.
Even if it's a true partnership where they produce on behalf of you with your criteria as a dedicated line?
No. I mean, I say no, but it's not that decided yet. Up to now, we want to master the entire process. But of course, if we find a partnership, I mean, I don't, as we always say with Mr. Barisson, we don't want to reinvent the wheel. If we have a good partnership that already masters all those techniques, probably we will go for a partnership. Yeah.
So another question from our friend Jean-Christophe, and hello Jean-Christophe. Do you have the DMPK data about your molecule, the drug metabolism and pharmacokinetic? Sorry, not yet.
Not yet. So we have it because we've done a human clinical trial on another application. It was not brain trauma, it was depression. And we have a lot of rat data with DMPK, pharmacokinetics, and so on, but not at the pharma level yet. Not at the pharma level yet. I have done a first fundraising that will allow me to get these kind of data before finishing the. Actually, with this fundraising, I will have the ADME, the absorption, distribution, metabolism, excretion, PKPD, and I will need an extra round of financing, the 6 million I was talking about, to go for the toxicological part. I don't have them yet.
Okay, next question, Chris. Hello, Chris. I guess we answered this question, on the origin of the raw material and would it be possible to collaborate? So you've got the answer, not so far. Next question is about could MSP be used to considerably reduce the antibiotic used in fish aquaculture? Huh? That's a good question.
It does. It does. It's amazing. So this is a more Olmix topic because it's dealing with animal care. But if you want to have this discussion, we can connect and discuss about it. I know that the group is already using the MSPs in the fisheries, and there are two projects really interesting regarding this question. The first one is always the boost of immune system to avoid use of antibiotics. In Norway, they are of course really interested in this project. And the second thing is that I have met a team that have engineered algae that that take antibiotics in the water and transform it, metabolize it, and transform it into secondary metabolites that are not antibiotics at all. So they have developed this kind of algae filter that just takes antibiotic from the water and transform it into and destroys it. Actually, it's just destroying it. So algae, I think, is really one very good tool to tackle those two questions. And if you want to discuss about this in more details, please let's connect.
I will connect everybody afterwards, no problem.
Nice.
Another question from Jax in the main. Hello, well, welcome, to the BioMarine online session, and even our friends, from US are present. So what is the competitive advantage of the ULVA MSP versus, the sulfated biopolymers, and how they compare and how can they be protected by patent? I guess we all have, part of the answer, but it would be interesting to get yours.
Yes. So patents, we already have the patents on what I just presented, and they are about to be delivered. And so I have no real issue about the patents because it's not
a natural ingredient cannot be protected. So there is some technology, there is some
some technology, there are some processes, and there are some exact way of doing things. You cannot just take the algae and try to make an extract. There are 25 years of R&D in the Olmix group to get this molecule. And if you look at the industrial process to get the MSP, this is protected, and it's not so easy. It's really not so easy. It's difficult to replicate. So, and this is why also Olgram is using the Olmix way of doing the MSP. So this is to answer the patent part. And by the way, it's a very good negative control in our experiments. We buy MSPs or we extract MSPs in another way, and it's not working. So I know I'm just pushing the questions a little bit further because your next question will be, but why this one is working and not the other one? This is the $10 million question. We are studying it now, and it's a very good tool to have something working, something not working. We are doing omics, genomics approaches, and we have very good, very interesting data starting to show us why MSPs are working. But bear in mind, those three properties. Yes, there is an immunostimulation, but in the end, if you take a leaf of a tree, you cut it, you inject it in your blood, you will have immunostimulation. So it's barely the point in the immunostimulation. So you have immunostimulation, but bear in mind, if you take an animal, you inject high doses of MSP, you have no immunostimulation. When you have an immunodepression, you do have an immunostimulation. And last but not least, and this was not expected at all, and this is something that is also patented, and it's serendipity. We're not expecting that at all. If you have bacteria in the lung, after 24 hours, they will be everywhere. They will disseminate. It's going to be a septicemia. But if you have bacteria in the lung and you inject the MSP, the bacteria stay in the lung. I mean, why? Why? We start to know why. But this was specifically patented. And those 3 points, when patients have a trauma and they have a tube in their mouth, and that's where the pneumonia enters. Sorry to talk like that, but that's where the pneumonia enters. Well, if the bacteria is in the lung via the tube in the intensive care units, if the bacteria stays in the lung, it's much easier together with the immunostimulation to get rid of it.
So sorry for my mistake, Jax, because, Eva, I didn't recognize you, but Eva is the next lady that will be on the hot seat, next month. And she's a specialist of Caragi, and she's already, producing some spray, antiviral spray. So I think I'm going to connect you with Eva. I guess you have some very good comment.
Absolutely.
Pierre, For our audience, would it be possible to share the presentation afterwards on our website?
Yes, of course. Yes, yes,
without
yes.
any
My pleasure.
information?
Yeah,
Okay.
my pleasure. Yeah.
Yeah, perfect. Let me just see if we don't have answered this type of, so we, oh, Eva is asking, that's a cool one, if there's any similarity of MSP to heparin or heparin sulfate.
Yes, there are a lot. It's well, it's a sulfated polysaccharide. So in terms of chemistry, they share a lot of common features. And we are collaborating with a pharma company who knows a lot about heparin, by the way. However, heparin was one of our very first positive control in all our experiment. And for example, the anticoagulant effect of heparin, we do not find it in our MSP. And this was really important because you don't want anticoagulant properties in a traumatized bleeding patients.
Yeah.
So that was something very, very important. And also what is also very important is that at very high doses, heparin is pyrogenic, which means that it triggers fever. This is something that cannot happen with MSP. We tested it, it's not happening, and it's good that it's not happening because a patient at the hospital with a fever means that he has an infection and the doctors will be not willing to use our molecule if it triggers fever in the patient.
So the final product will be oral or injectable?
Injectable. IV. We want something that acts very fast and well, patients with a tube at the hospital and so on, they have the connection with the IV connection already, so it's easy to add a molecule IV.
And Jean-Christophe is asking about the effect on the microbiota. Do you think that there might be when you inject it? Do you think that there might be some incompatibility with the microbiota, or would it be fitting well, not causing too many effects?
So much less than an antibiotic for sure, a broad spectrum antibiotic for sure that will cause much less effects. However, I am 100% sure that we actually have data showing that the microbiota is affected by MSP. When I told you before that we had an interesting effect on depression, most of you probably know that there is a link between depression, the immune system, and the microbiota. If you take the microbiota of a depressed mouse and you inject it in a healthy mouse, the healthy mouse will become depressed. So the microbiota is very, very important. And this is probably our mechanism of action. I say probably because we don't really know. Microbiota for the MSP in the depression experiments. So are we going to have an effect on the microbiota? Yes, but to be honest, someone who is at the hospital after a traumatic brain injury, generally they stay 2 weeks to 3 weeks. They are fed with, the tubes. They take a lot of drugs. I don't think that MSP will really count in the modification of the microbiota, not to mention the stress and so on. So yes, we will have an effect. And I think it's a good topic for the Bowel syndrome subject. But in the case of traumatic brain injury, I don't think it's really an issue.
Quickly, Pierre, because we just have 2 minutes before we close.
Yes.
Down, and you were talking about the others molecules. Is there any peptide or proteins that you're going to use in your drug candidate, or is it combined, or is it still the MSP?
No, it's so on the one hand we have the MSP, and you know a lot about it now, and we have the other program which is a peptide. This one is much easier because we can synthesize it completely in a factory. We can buy the molecules that are extracted from the sea. And this other program is also very, very exciting because we are the only one able to kill sleeping bacteria that are responsible of chronic infectious disease. So this is also an amazing result. And this is something we are also pushing. And we think at Olgram that in order to fight against antibiotic resistance, you boost the immune system, but you also need new tools to kill the bacteria.